Good morning. My name is Li Long Huang. I'm from Emergent Biosolutions. I want to ask, do a little survey, and please show supported by your raise of hand. Have any of you heard about next generation bioprocessing? How many of you? How many of you have heard about multicolumn chromatography or continuous chromatography? How many of you have heard the implementation of multicolumn chromatography in a commercial process? That is what Emergent is trying to do. So today's topic is a roadmap to the licensure for multicolumn capture in a map process. Right. First of all, I'd like to acknowledge the team from Emergent and the management team and my colleagues for the support and the list people, and also like to thank the Paul team for the execution of the studies. And some of the names you might find familiar, Mark Bishop among the Paul team. So the agenda I will share with you a little bit about the Emergent Biosolutions, and then I will delve into the next gen vision of for Emergent, and also talk about the pilot of multicolumn chromatography, and then summarize who we are. Our mission is very simple: to protect and enhance life. As a global life science company, Emergent is focused on providing specialty products for civilian and military populations that address accidental, deliberate, and naturally occurring health threats. That is what Emergent attracted me to join the company. Emergent has 19 global locations, 11 market products, 15 pipeline products, and 4 platform technologies, and multiple CDMO business services. And there's a variety of services that Emergent provide. Please stop by at the booth 2961 to learn more about the service that the company offering. I want to especially talk about the Bayview facility. We have a Center of Innovation, Advanced Development and Manufacturing facility. That's where I'm from, and this is one of the 3 centers in the U.S. and is a public-private partnership with BARDA. We provide surge capacity ready for infrastructure for biologic ready MCMs. So for the biologic process that Emergent is trying to work is achieve, as any of the biological manufacturing companies trying to do, to achieve flexibility, achieve consistent product quality, achieve speed to market, and reduce cost. How are we going to do it? We're trying to achieve, looking at 3 pillars. That's single use, continuous manufacturing, and process analytics. Emergent is implementing 4,000 liter single-use bioreactors for a commercial map process. That's where we are for a single-use platform. And we believe to move to the next steps, we need partnership with a variety of industrial leaders and also regulatory agencies. In terms of the continuous processing, that's where this topic is more about. And our platform is based on single-use platform. And our strategy for drug is focused on drug substance at the moment from end to end. The strategy we took is leveraging the continuous processing capabilities and also process intensifications from the industrial leaders and implementing those technologies that has more potential to implement it in commercial program. So the subjects could include multicolumn chromatography, perfusion, inline dilution, high-density cell banking, and also the integrated downstream processing. Now move on to the multicolumn chromatography. Why multicolumn chromatography? Let's maybe talk about how the multicolumn chromatography works. That in the batch processing, you only have one column. In the multicolumn chromatography stage, you can load the column. In this case, I'm looking at a 3-column scenario. The product will be loaded on the first column. the breakthrough will be collected in the second column, while in parallel, the third column is being prepared, ready for the loading again. So when the first column is loading completed, you move on to the next column loading, where the first column is being eluted and regenerated, ready for the loading again. So that's how it works. In the end, the product is feeding to the multi-column system continuously, and the product is eluted from the outlet continuously. The right shows the elution profiles, multiple cycles. So going back, why Emergent interested in the multi-column chromatography implementation? Of course, cost reduction is one of the major drivers. Secondly, multi-column chromatography has been in existence for more than a decade. A variety of industry companies has explored the capabilities. It has demonstrated that it can reduce the resin cost and also enable process flexibility. For us, because we're using a single-use platform, this fits our application really well. And one of the drivers why we're looking at multi-column chromatography is because multi-column chromatography has been in existence for 10 years, has been explored by a variety of industry leaders, and it's mature enough to take it to commercial implementation. Therefore, Emergent is partnership with Paul, where Paul has this leading technology, multi-column chromatography, with established collaboration. To make this in reality, we took a phased approach. Phase number one, we ask the question is it feasible to convert from batch to continuous? How complex it is to convert to continuous? Afterwards, then we plan to implement it to large-scale implementation and really take to licensure. How the implementation works, how the design works, and first of all, we do multiple break curve studies, and kudos to Paul, they executed all the studies for us. They run the break curves at a variety of flow velocities. Here we're looking at 300 centimeters per hour, 600, and 900 centimeters per hour. To put this into perspective, if you look at Cephros columns, the flow rate is around 100 centimeters per hour. Typical protein columns around 300 centimeters per hour, where this high flow rate trying to increase the throughput. That's one of the major drivers for multi-column chromatography. So in this case, we identified the static binding capacity, and from that, the process phases for the multi-column chromatography, how many columns is being evaluated, designed, and what the flow rate is selected. In the end, we selected a flow rate at 700 centimeters per hour. We select 3 columns for this operation. How the process performs is that design works. This is the multi-cycle elution profiles, and there's no, in between cycles, there's no peak, and there's no overlay. And this shows that the process design, phase design is robust. We can move on to the next steps. Right? The next question is, how was the process performed in terms of recovery, in terms of product quality? We look at the batch processes, we look at the continuous mode performances. The product yield is the product yield are comparable, 100% for both batch and the continuous, where the impurity clearances, including the HCP and the DMA removal, are comparable, and the aggregate levels are comparable. So what's our next step? Right? With the proof of concept feasibility is being implemented, we plan to the next step is we're in the discussion with look at the cost reduction and working with Paul on the cost of goods for the assemblies. The next step is really look at the risk mitigation before we go to large-scale implementation. Look at the bioburden management, look at the viral clearances, look at the scale-up effect. And most importantly, we want to look at the process robustness and in terms of the comparability from batch to continuous mode. And after that is done, we'll move on to the commercial implementation. And throughout this process, we don't believe that we can do it all alone by ourselves. We have to, just like you probably heard Amgen's experiences, heard about Vertex experiences, implementing continuous process for small molecules. It takes multiple years. And their recommendation is really to partnership, right? Partnership with industrial leaders, partnership with agencies to get the feedback earlier. That's what we're asking, is to work with those parties and to design a pathway to real commercial implementation. To summarize, in this talk, I shared with you what Emergent's next generation bioprocess vision is. We're focusing on 3 pillars: single-use, continuous processing, and process analytics. And in terms of the continuous processing arena, we're piloting the multicolumn chromatography and putting on a map, commercial map process. Our goal is to implement that for the map process. And we're partnership with Paul in this implementation. And we're looking forward to working with the agencies to develop a pathway, regulatory pathway, to make this a reality. Thank you.
Wei Wang, thank you very much. We do have time for a question or two in the audience, if we have any. If not, oh, got one.
Thank you for your talk.
Thank you.
A couple of quick questions. I didn't really understand the scale of the experiment. Are you talking about just running a few liters, and also the length? So it seemed like it was 20 hours, and yet you're intending to integrate with perfusion, right? So can you comment on those two things?
Thanks for the question. I think right now the first phase is really looking at implementing for batch process. So the upstream will be, right, the scale is about 4,000. So we implement the continuous mode to processing the feedstock, and in terms of volume will be 4,000. Yeah.
Great, thank you. Any other questions? Yes.
Thanks. I was wondering, how close are you from any kind of cGMP manufacturing with this method, even if it's at small scale?
Could you repeat that question?
So I wonder if, are you close or far in terms of years to any kind of cGMP manufacturing using this for even small scale?
I don't think the small scale has the capability, the small scale devices here is ready for cGMP. You have to go to a process scale, and our plan is, I think we'll do scale pilot studies around that scale not in GMP. Then eventually that's why we want to take it to GMP implementation, and using the 4,000 liter scale and process it through the process scale equipment, and then evaluate the scale-up feasibility, and then implement it, do the PPQ, whatever, do comparability as well, and licensure. So we are planning to do the cGMP studies. Yeah.
Thank you very much for that question. Any other questions? Wei Wang, thank you very much.